Good day, GSLSAMP community. My name is Justin Durant, and I'm a researcher at Kane University, here to present to you my research that was conducted at Kane University. The research was done on the bacterial microbiome and how it negatively affects agricultural plant growth. And it was funded through GSLSAMP and the School of Natural Sciences at Kane University. Everyone that worked on the project was me, Justin, Brianna Crowley, Shane McLeod, Lucy Paul, Yanelis Rivero, Christiana Wu, and of course Nicholas LaRusso, who was our professor and really guided us through the whole thing. Um, so to start off, invasive plants are uh, are being introduced into ecosystems more and more. These invasive plants have been known to influence soil microbiomes that they're introduced to. These species could disturb the native microbiota, or even introduce others. Native plants rely on natural soil bacteria in order to support their growth, as well as helping them carry out processes like nitrogen fixation. Invasive species could affect the growth of agricultural crops that are important to the industry. In order to evaluate the effects of what could happen to these crops, we used bacteria that was collected from areas with two invasive species, and we collected ones from areas without. These samples were then inoculated into soil that contained tomato seeds, and they were then monitored to see how that affected their growth. So, summary of that, um, what that's pretty much saying is that soil is a complex community of bacteria that are mutualistic and affect each other. The best way that I can like that too is it's similar to like a food web, where the addition or subtraction of one microbe could adversely affect how a plant grows because they're all intertwined. So what we did was we collected soil from areas, 5 different areas that we knew had invasive species, and samples from 5 areas that we know were native, and how it in turn should be. um, our method section down here. The soil was collected from tomato plants. Um, Soil samples were then analyzed and tested for invasive species. Each student was given a different soil sample at the beginning of the semester, and each was eluted. Well, we eluted each of the samples to isolate the DNA, and PCR prep was done to make copies of the DNA, the bacterial DNA that was present in the soil samples. Measuring the tomato plant lengths allowed us to get estimated heights each week so we can analyze that and see how that relates to our thesis. We then used light field microscopy to observe native and invasive bacteria. The bacteria was cultured using PDA plates, and these plates were able to, and with these plates, we were able to isolate different colonies of bacteria. So again, what that means is, or what happened was, we created soil slurries. We added water to the soil samples that we collected in order to it's like separate the bacteria from the soil, and that made it easier for us to then take our PCR tests and culture it on our plates. The light field microscopy is while it looked good, to see the microbes culturing or growing on the plates. It was really the PCR test that gave us a very accurate description of all the microbes that were present in the soil. Not all microbes grow on plates, so it was not as nearly as accurate as the PCR test that we conducted. Our results was that the average plant height for the tomato plants um, inoculated with bacteria associated with invasive plants was notably lower in the by the last week of our observations. Um, Both environments had a similar overall pattern, but the maximum size obtained for the plants with the bacteria associated with the invasive plants was significantly different. So what that is showing is that as you see every week they did grow around this, like each plot grew around the same height, but the ones that did have the invasive bacteria only grew to around 11 to 14 uh 14 centimeters, and that's the highest they got, while the ones that were left with their native microbiota grew to 20. The highest the max height that we got for those was 20 centimeters. And to conclude, essentially, and what that meant to us was that based on our results and our analysis, we found that non-native microbiomes do adversely affect tomato plant growth. Tomato plants inoculated with foreign and invasive microbiomes grew roughly 50 centimeters less than the controlled tomato plants with the native microbiota. So in total, if you averaged all the plants from the native compared to the invasive, the invasive plants, or the tomatoes with the invasive microbiome grew 50 centimeters less in total than the ones that were native. And uh yeah, that was fairly alarming. That's something uh definitely did want to highlight. Samples showed that there was an addition of foreign microbiota in the soil samples with the invasive plants present. And we really found that out through our PCR prep. Future research, we believe, should be done on the mechanisms in how these microbiota really affect the growth. We know it does change the overall microbiota, like it added microbes to the total ecosystem, but we don't know exactly why and how that affected the growth. So it's definitely something we couldn't finish in one semester, but definitely something worth looking into. It can be concluded that the lack of growth is likely due to the change in the microbiology of the soil. And that's our research. All our references are down here. And yeah, I appreciate your time, and I hope you guys have a nice day.